day, everyone. I am Ma'am Maricar A. Ganas. In today's lesson, we'll explore the fundamental molecules of life, the DNA and RNA. These molecules are essential for they carry genetic information and enabling synthesis of proteins, which are crucial for the structure and functions of all living organisms. In this lesson, we'll examine the structures of DNA and RNA, their functions, and how they interact to cellular processes. By the end of this lesson, you will have a clear understanding of DNA and RNA and how they play in genetics and molecular biology.
Let us begin with DNA, or deoxyribonucleic acid. DNA is the molecule that carries the genetic blueprint of all living organisms. It is responsible for storing and transmitting the genetic information required for cell function, growth, and reproduction. The structure of DNA is known as a double helix, which resembles a twisted ladder. The rails of this ladder are made up of sugar molecules, specifically deoxyribose and phosphate groups. The steps of the ladder consist of nitrogenous bases. There are 4 types of nitrogenous bases: adenine A, thymine T, cytosine C, and guanine G. These bases pair in a very specific manner. Adenine pairs with thymine, and cytosine pairs with guanine, forming what are called base pairs. The structure of DNA is crucial because it allows for easy replication. When a cell divides, the DNA molecule unzips, and each strand serves as a template for a formation of a new strand. This ensures that genetic information is accurately passed on to the next generation of cells. Now, let's move on to RNA, or the ribonucleic acid. DNA, like RNA, is a nucleic acid that is essential for protein synthesis. However, there are key differences between DNA and RNA. RNA is typically single-stranded, whereas DNA is double-stranded. Additionally, RNA contains a different sugar called ribose, while DNA contains deoxyribose. Another difference is RNA contains the base uracil or U instead of thymine or T, which is found in DNA. RNA comes in different types, but the most important for protein synthesis are mRNA or messenger RNA, tRNA or transfer RNA, and rRNA or the ribosomal RNA. Now, let's take a closer look at each of these. First, let us try to have a closer look for the messenger RNA or the mRNA, which is the molecule that carries the genetic information from DNA in the nucleus to the ribosome, where proteins are synthesized. It serves as a template for protein production. Next is the tRNA or the transfer RNA, brings the appropriate amino acids to the ribosome during the process of translation. Each tRNA molecule has an anticodon that matches with a specific codon in the messenger RNA, ensuring the correct amino acid is added to the growing protein chain. Finally, the rRNA or the ribosomal RNA is a key structural component of the ribosome, the machinery that reads the messenger RNA and assembles the amino acids into proteins. Let us summarize the key differences between DNA and RNA. First, for the strands. DNA is a double-stranded, while RNA is single-stranded. Second, for the sugar. DNA contains deoxyribose, whereas RNA contains ribose. Third, for the bases. DNA uses thymine, while RNA uses uracil. Fourth is the function. DNA stores and transmits genetic information, whereas RNA is involved in protein synthesis. Fifth is the location. DNA is primarily located in the nucleus, whereas RNA is found both in the nucleus and in the cytoplasm. In conclusion, DNA and RNA are both critical molecules in biology, and they work together to ensure the accurate transmission of genetic information and the synthesis of proteins. DNA provides the genetic code, while RNA plays a key role in interpreting that code and facilitating the production of proteins that are essential for life.
Thank you for your attention, and I am looking forward to be with you in the next session.